Okay, let's unpack this. Imagine the power to precisely rewrite the very code of life, our DNA. It sounds like, well, straight out of science fiction, doesn't it?
It really does.
But with revolutionary gene editing techniques like CRISPR, this is fast becoming a tangible reality.
Yeah, interestingly, the pace of progress in this field is just breathtaking. I mean, just a few short years ago, the idea of routinely editing genes felt like a far-off aspiration.
Right.
Now it's at the very forefront of scientific and medical innovation.
Exactly. And that's precisely what we're diving deep into today. You've pulled together a really fascinating collection of articles that sheds light on this incredible world of gene editing, our mission, to sort of extract the crucial insights, understand the real-world impacts, and explore the significant considerations surrounding it, all in a way that's clear, engaging, and doesn't feel like you're deciphering a genetics textbook.
Yeah, and if we connect this to the bigger picture, you'll see that this isn't just some highly technical discussion. It really touches upon fundamental aspects of our health, how we grow our food, even our relationship with the environment. It's a field just brimming with potential for positive change, but, one that rightly calls for careful thought.
So where does this incredible story even begin? It turns out the foundation for CRISPR was laid by pure, old-fashioned curiosity. Tell us about Francis Mojica and his work with some seemingly, ordinary bacteria.
Yeah, it really raises a fundamental point about how scientific discovery often unfolds, doesn't it? So back in the 1990s, Francis Mojica was studying bacteria thriving in salty soils. These weren't, bacteria causing major epidemics or anything particularly glamorous.
Right.
But he noticed something peculiar. Their genomes contain these repetitive DNA sequences. Just odd. And his curiosity about this oddity, this apparent defense system in these, quote, inconsequential bacteria, well, that ultimately paved the way for a scientific revolution. It's just a powerful reminder that fundamental research driven by simple curiosity can have profound, totally unexpected consequences.
A genetic vaccination card. That's how he described it, right?
Right.
Explain that fascinating concept.
Precisely, yeah. Mojica realized that these repetitive elements called CRISPRs were actually interspersed with short sequences of DNA that perfectly matched the DNA of viruses that had previously infected the bacteria. It was as if the bacteria were keeping a genetic record of past viral attacks, almost like a microbial memory bank.
Wow.
Which allowed them to recognize and fight off those viruses if they encountered them again.
And these records weren't just sitting there idly. They had a specific function.
Absolutely. Associated with these CRISPR arrays were genes called Cas genes, which produced Cas proteins. These proteins, guided by RNA molecules transcribed from the CRISPR sequences, could specifically recognize and cut up the DNA of invading viruses.
Cut it up.
Yeah. It was a remarkably sophisticated system for identifying and neutralizing threats right at the genetic level.
It's amazing to think that scientists in the dairy industry were also early observers of this. Yogurt and cheese production, who knew it, held vital clues to gene editing.
Yeah, it's funny how seemingly unrelated fields of research can converge, right? Scientists in the dairy industry were struggling with viruses that could infect and just decimate their bacterial cultures.
Oh, right. For yogurt and cheese.
Exactly. Essential stuff. They observed that some bacteria seemed to develop resistance to these viruses over time, and eventually they connected this resistance to these CRISPR systems. They realized the bacteria were adapting and protecting themselves using this mechanism.
So fast forward to around 2011, and we get this pivotal collaboration between Jennifer Doudna and Emmanuel Charpentier at Berkeley. Their focus, this particular Cas protein called Cas9.
Right, and here's where it gets really interesting. Doudna and Charpentier, they were driven by a fundamental question about, the precise function of Cas9, so they joined forces. And their groundbreaking research revealed that Cas9 was basically the workhorse of this bacterial immune system. Think of Cas9 as molecular scissors, guided by a piece of RNA that matches the exact DNA sequence we want to edit. This guide RNA, it's derived from those stored viral DNA sequences in the CRISPR array, allowing Cas9 to pinpoint and slice specific DNA targets with just incredible accuracy.
So it's like a guided missile for DNA. Break down the key components for us a bit more.
Yeah, you could think of it that way. Imagine having a very precise GPS system that's the guide RNA that leads a pair of molecular scissors, Cas9, to a specific location within this huge map, the genome.
Okay.
The guide RNA contains a short sequence that perfectly matches the target DNA. Cas9 then unwinds the DNA double helix, and right at the precise location specified by the guide RNA, it makes a double-stranded break.
A cut.
Yeah. Initially, they also identified another RNA molecule, the tracer RNA, which helps the guide RNA bind to Cas9. But later, they ingeniously combined these two RNA bits into a single guide RNA.
Simplified it.
Yeah, significantly simplified the system, made it much more user-friendly for gene editing.
And this is the crucial leap, right? The realization that by making these targeted DNA breaks, you could essentially hijack the cell's own repair mechanisms to introduce specific genetic changes.
Exactly. That was the unexpected transition. From understanding a bacterial defense mechanism to realizing its immense potential as a gene editing tool for, well, virtually any organism.
How did that work? The hijacking part.
So when Cas9 makes that precise cut in the DNA, the cell recognizes this damage and tries to repair it. There are two main ways it does this. One pathway often introduces small insertions or deletions right at the cut site, which can effectively disable or knock out a gene. But the other pathway, if you provide the cell with a template of DNA alongside the CRISPR-Cas9 system.
Like a replacement piece.
Exactly, a template.
Yeah.
The cell can use that template to repair the break, allowing you to insert a new gene or replace an existing one with a corrected sequence. That's the fundamental principle behind precise gene editing.
Okay, wow. Now, while CRISPR-Cas9 rightly gets a lot of the spotlight, it's important to remember it's not the only tool in the gene-based therapy toolbox, is it?
That's a really crucial point to keep in mind, yeah. While CRISPR has, understandably generated tremendous excitement, the field of gene-based therapies is actually quite broad and keeps evolving.
Like what else?
Well, we have approaches like antisense therapy, where drugs like Spinraza target RNA molecules to correct gene expression. That's used for spinal muscular atrophy.
Right.
Then there's RNA interference, or RNAi. Drugs like Onpattro use that to silence specific genes at the RNA level for conditions like hereditary transafferrin-mediated amyloidosis. And of course, there's traditional gene therapy, which involves introducing working copies of a gene into cells. That's shown promise, too.
And now with CRISPR, we have this, unprecedented ability to directly rewrite that faulty genetic code. Like what's being explored for Kabuki syndrome. It must be offering families facing these single-gene diseases a much wider spectrum of hope.
Oh, absolutely. For so many families grappling with the challenges of single-gene diseases, the landscape of potential treatments has just expanded dramatically. They're not limited anymore to just traditional gene therapy or protein-based approaches.
Right.
The arrival of CRISPR gene editing offers this real possibility of more precise and hopefully more effective interventions that could actually address the underlying genetic cause of their condition.
Let's delve into some of these real-world applications then. We're already seeing gene therapy making significant strides. The first FDA approval was back in 2017, right?
Yes, the field has been steadily building momentum, and now gene editing is demonstrating just incredible promise across a diverse range of conditions. Take Wiskott-Aldrich syndrome, WAS.
Okay.
It's a rare and serious immunodeficiency. Research led by Dr. Matthew Porteous suggests that CRISPR could allow for a much more precise insertion of a corrected gene compared to older methods.
Getter targeting.
Exactly, potentially leading to improved gene expression and a more effective, lasting treatment. There's, considerable optimism that clinical trials for this approach could begin within the next, say, 1 to 3 years.
And we've seen some major milestones just very recently, haven't we?
Yeah.
The federal government's approval of a gene editing treatment for sickle cell disease. That's a truly significant event.
It is a landmark moment, absolutely. And this progress isn't just in the United States. Over in the UK, NICE, that's the National Institute for Health and Care Excellence, they've recommended Casgevy, a CRISPR-based therapy for beta thalassemia.
Casgevy.
Yeah. And this recommendation means that potentially around 460 patients, aged 12 and over, in England and Wales could benefit. What's also particularly exciting is the prospect of India developing cost-effective CRISPR-based treatments for sickle cell anemia and beta thalassemia.
Which affect huge numbers there.
Exactly. Conditions that unfortunately affect millions of people in that region. The potential for global impact is really enormous here.
What about other single-gene diseases? Things that have historically been so challenging, like Huntington's, liver diseases, Duchenne muscular dystrophy.
Yeah, research efforts are very active in those areas, too. For instance, Dr. Eric Olson's work in dogs with Duchenne muscular dystrophy has shown some really encouraging results.
In dogs?
Yeah, using gene editing techniques to alleviate some of the disease's effects. The ability to precisely target and correct the underlying genetic defects offers a potential therapeutic path for many of these debilitating conditions where effective treatments have just been elusive.
It's not solely about treating diseases in living individuals, though. I read scientists are even exploring using gene editing to modify pig organs for human transplantation. That sounds, well, revolutionary.
It really does. That's another fascinating application, tackling the critical shortage of organs for transplants. Scientists are using CRISPR to address the two main challenges in xenotransplantation.
Which are?
The potential for pig viruses transmitting to humans and the rejection of pig organs by our immune system. So, by precisely editing the pig genome, removing these viral sequences, and making the organ tissues more compatible, more human-like, they're hoping to overcome these big hurdles and make xenotransplantation a viable option.
Wow. And moving beyond direct therapies, CRISPR is also proving valuable in diagnostics.
Absolutely. The inheritability of CRISPR-Cas systems to precisely recognize specific DNA sequences is being cleverly harnessed. They're developing highly sensitive and accurate diagnostic tools.
So faster detection.
Potentially, yeah. Detecting disease-causing DNA much earlier, which could lead to more timely and effective disease management.
The applications seem almost to stretch the imagination. I was really struck by the research at UCSF using modified CRISPR-Cas9 injected directly into mouse brains for neurodegenerative diseases, even cancer therapy.
Yeah, this really highlights the remarkable versatility of the technology. By carefully modifying the Cas9 protein and refining how they deliver it, researchers are exploring innovative ways to reach parts of the body that were previously really hard to access, like the brain. This opens up exciting possibilities for targeting the underlying mechanisms of complex conditions like Alzheimer's or Parkinson's, and for delivering cancer therapies right to the tumor site.
Okay, let's shift focus a bit now. Let's discuss the huge impact CRISPR is poised to have on agriculture. We're hearing about creating crops with enhanced resistance, better nutrition.
This is an area where CRISPR holds truly transformative potential, especially on a global scale. I mean, imagine engineering crops that are more resilient to drought.
Which is increasingly important.
Definitely. Or resistant to devastating diseases and pests, or even enhanced in their nutritional content. This could really revolutionize food production, improve food security, and maybe reduce our reliance on some traditional farming practices that have, environmental downsides.
Zach Lippman's work with tomatoes, using CRISPR as a rheostat to control fruit production. That sounds incredibly precise.
It truly is. By precisely targeting specific regulatory genes in tomato plants, Lippman and his team showed they could finely tune the number of fruits a plant produces. That level of control could significantly boost crop yields.
And then there's that fascinating example, the non-browning mushrooms from Penn State. A small genetic change, big potential for reducing food waste.
Exactly. Using CRISPR to specifically knock out a single gene responsible for that enzymatic browning.
The one that makes them go brown when you cut them.
Yeah. They've created mushrooms that keep their fresh appearance longer. It's a simple application, but it perfectly illustrates CRISPR's precision and its potential to solve really practical problems in food production and consumption.
It's interesting, though, seeing the different regulatory approaches. The U.S. seems more permissive with CRISPR-edited crops than Europe, for instance. What are the implications there?
Yeah, this divergence has significant implications for international trade and just how widely these technologies get adopted. In the U.S., the USDA generally doesn't regulate CRISPR-edited crops if no foreign DNA is introduced. They often see these changes as similar to what you might get through traditional breeding.
Okay.
But the European Union tends to classify them as GMOs, genetically modified organisms, which means much stricter oversight.
Right. So that creates issues for global markets.
It does. Complexities for global markets influences consumer acceptance, different standards, different labels. It also highlights this ongoing debate about how we define genetic modification. Is it the final product or the process used?
And the fact that CRISPR can potentially offer these sort of natural-like edits without foreign DNA really sets it apart from earlier GMOs, like Blade cotton.
That's a critical distinction, yeah. Traditional GMOs often involved inserting genes from other species. With many CRISPR applications in agriculture, you're making very precise edits to the plant's own existing genome, no foreign DNA integrated. That raises interesting questions about categorization and regulation.
Let's transition now to the environmental side. The concept of gene drives in particular sounds like it holds both incredible promise and, well, potential for concern.
Gene drives definitely represent a particularly powerful application of CRISPR with the potential for significant ecological impact. They work by using gene editing to essentially force a specific genetic trait to be inherited preferentially.
So it spreads really fast.
Exactly. Spreads rapidly through an entire population over generations. One of the big applications being explored is controlling mosquito-borne diseases like malaria and dengue. The idea is to use gene drives to make mosquitoes unable to transmit the disease, or even make them sterile, reducing their populations. There are ongoing field trials and tests happening in Africa and the U.S.
But the potential for unintended ecological consequences, that seems like a really significant concern that needs careful thought.
Oh, absolutely. Because gene drives are designed to spread through entire populations, any unintended ecological effects could be very difficult, maybe impossible, to reverse. So that demands a very cautious, thorough approach, comprehensive risk assessments, careful consideration of impacts on complex ecosystems. We have to tread carefully there.
Beyond gene drives, are there other potential environmental uses for CRISPR being looked at?
Yes, researchers are investigating a range of other possibilities. Things like engineering bacteria to break down pollutants in contaminated rivers more effectively.
Okay.
Or enhancing the efficiency of algae or other microorganisms for biofuel production. Again, these hold promise, but they also need careful research and understanding of potential unintended consequences before any large-scale rollout.
This brings us to the really profound part of our discussion, the ethical and societal considerations. I mean, we're talking about the power to manipulate the very code of life.
It truly is profound. We're gaining the ability to alter the fundamental building blocks of life, potentially influence the evolutionary path of organisms, including ourselves. And that naturally raises huge ethical questions about our role, our responsibilities, the potential outcomes, stuff we really need to grapple with as a society.
The distinction between somatic cell editing and germline editing seems particularly important here. Can you clarify the difference and why germline editing tends to cause more concern?
Sure. So somatic cell editing involves making genetic changes to the non-reproductive cells in an individual's body, like liver cells or blood cells. These changes affect only that person and aren't passed on to their children.
Okay, not inherited.
Right. Germline editing, on the other hand, involves making changes to eggs, sperm, or embryos. These changes do become part of the individual's DNA and can be inherited by their offspring, potentially altering the human gene pool for generations.
Wow. So the mouse egg example earlier, injecting editing molecules, that's germline.
Exactly.
Right.
That leads to heritable genetic changes.
And it's this heritable aspect of human germline editing that understandably fuels a lot of the discomfort concerns about potential eugenics, widening societal inequity.
Precisely. The prospect of intentionally altering the human germline raises serious ethical red flags about potential eugenics, selectively breeding humans for certain traits.
Right.
There are also significant worries about access. Who gets these technologies? Could it widen existing inequalities, creating a kind of genetic divide? And the fundamental questions, who decides which traits are desirable for what purpose? These are deeply complex issues.
The case of Heijiankui in China, the babies born with edited genomes, that really brought these ethical issues into sharp, urgent focus globally, didn't it?
It was deeply concerning, yeah. Widely condemned by the scientific community and beyond. Jiankui claimed he edited the CCR5 gene in human embryos to give them HIV resistance.
But the methods?
The methods, the lack of transparency, it all raised serious ethical and safety concerns.
And the actual genetic edits he made were novel, untested. That seems incredibly risky.
Exactly. The specific changes he introduced weren't well understood. Their potential off-target effects, long-term health consequences unknown. These edits hadn't been rigorously tested in animals or human cells before being used in embryos.
Wow.
That lack of scientific rigor, the potential for unforeseen health problems for those children, it sparked widespread outrage. It really underscored the critical need for clear ethical guidelines and strong regulatory oversight. As one prominent scientist said at the time, it made the hairs on my neck stand up.
That event really spurred international efforts to grapple with these complex ethical and regulatory challenges of human germline editing.
Yes, it was a stark reminder of the potential implications. It prompted organizations like the WHO, national science academies, to convene expert panels, start discussions, try to develop ethical frameworks for responsible innovation.
And there's ongoing debate about moratoria, like pausing certain applications.
Yes, There's a global debate about whether to implement moratoria on certain uses of human germline editing until we have a much better grasp of the risks and benefits, and until there's broader societal agreement.
We also have to consider the dual-use potential, right? The risk of bioterrorism, misuse.
That's another critical ethical dimension. Like many powerful technologies, CRISPR has dual-use potential. While it offers immense benefits, there's also the risk it could be misused, perhaps for developing bioweapons. There are also concerns, for example, in India, about potential misuse for non-therapeutic things like sex selection or skin lightening, which raises further ethical issues. Responsible development and strong oversight are absolutely crucial.
And even in agriculture, there are ethical considerations around corporate control, like Monsanto-Bayer, ensuring CRISPR serves people, not just profits.
Absolutely. Who controls the technology? Who benefits? We need to ensure that advancements in agricultural gene editing are accessible and benefit farmers and consumers broadly, not just concentrated in the hands of a few large corporations. Transparency and public good need to be priorities.
And altering the environment with gene drives, modifying animals, that needs careful ethical evaluation too.
Definitely. Any intervention that could permanently alter ecosystems or wild populations requires extreme caution and thorough ethical review. We need public engagement, input from artists, science communicators, everyone, to foster understanding and broad discussion.
It's also worth noting the rapid commercialization. The startups. Transparency about potential conflicts of interest for researchers seems important.
Very important. As researchers become involved in commercial ventures, maintaining transparency about potential financial conflicts of interest is vital for public trust and scientific integrity.
Which brings us to regulation. How do we strike that balance strict enough for safety and ethics, but not so stifling it kills innovation?
That's the million-dollar question, isn't it? It's a very delicate balancing act. We need regulations robust enough to ensure safety and prevent misuse, but flexible enough not to inadvertently halt potentially life-saving research and progress.
And it's global science, so international coordination seems key, but difficult.
Extremely difficult, but necessary. Science is global, so purely national regulations might not be enough. Achieving some level of international harmony in regulatory approaches is a major challenge, but one we need to work towards.
So what roles do the scientific community, journals, governments play in setting these rules?
It's a collective effort. The scientific community needs to self-regulate ongoing ethical discussions, best practices, transparency. Journals play a role through publication standards, and government bodies, regulatory agencies like the FDA, legislatures, they're essential for establishing the legal frameworks, the enforceable rules, ensuring accountability and public safety.
Like Senator Feinstein's interest you mentioned, showing policymakers are paying attention.
Exactly. That governmental attention highlights the growing recognition that we need clear, thoughtful regulatory requirements for this powerful technology.
And looking ahead, countries like India have a real opportunity here, don't they?
Yeah.
To potentially lead in ethical, responsible CRISPR innovation.
They really do. By establishing national initiatives, maybe fast-tracking approvals where appropriate, but with strong ethical oversight, encouraging public-private partnerships, investing in education, India could position itself as a leader in harnessing CRISPR's potential responsibly, especially for challenges like sickle cell disease that are so prevalent there.
So, wrapping up, while AI often dominates headlines about the future, it feels like CRISPR is very much the present for many transformative applications.
Precisely. AI holds immense future potential, no doubt. But CRISPR isn't just a future prospect. It's a present-day tool that's already reshaping medicine, agriculture, and our fundamental understanding of life itself. It's happening now.
So as our deep dive into this fascinating world of gene editing draws to a close, wow, we've covered an incredible amount of ground. From those initial curiosity-driven discoveries in bacteria.
Yeah, amazing starting point.
All the way to cutting-edge therapies and these really profound ethical debates shaping its future.
It has been a truly fascinating journey, just tracing the remarkable evolution of this powerful technology and exploring its multifaceted impact.
And that brings us to our final thought for you, our listener, to really chew on. With such a powerful tool like gene editing now readily available, what do you believe are the most critical considerations society must address to ensure its responsible and beneficial use for everyone? What specific applications fill you with the greatest hope? And which ones, if any, give you pause or raise concerns? We really encourage you to delve deeper into these important questions and continue this vital discussion.
Absolutely. The field of gene editing is just constantly, rapidly advancing. Thoughtful, informed engagement with all its implications is going to be absolutely crucial as we navigate its ongoing evolution and its increasing impact on our world.